everyone. Thank you for coming to join this webinar being sponsored by the Section of Genetics and Genomics for the American Thoracic Society. I'm Gloria Pryhuber. I'm at the University of Rochester in New York. I'm a neonatologist and a pulmonary biologist. I've been lead investigator for the Lung Molecular Atlas program, Human Tissue Core, and for the HubMap Lung effort. I do want to thank you all for coming. We're very excited by the attendance. It demonstrates how interested people are in this topic and how important it is that we get these discussions going. So without further ado, I'd like to just introduce the members of our webinar today. We'll have two speakers, Dr. Luecken and Dr. Scheuermann. Our moderators, myself and Dr. Martijn Nawjin from the University of Groningen. Our panelists, you'll meet in the breakout sessions, Xin Sun from UCSD, Gerard Koppelman from the University of Groningen. Jayaraj Rajagopal, I think actually isn't going to be able to make it today, but has been important in the planning of this session. Dr. Weillington Cardoso at Columbia and Dr. Nicholas Banovich at the Translational Genomics Research Institute in Arizona. So I'll give you a quick overview of the agenda for the webinar. I'm giving the introduction now, but we'll have two presentations, about 25 minutes each. The first one is on single cell data, reporting standards, cell identification and multimodality by Dr. Luecken. And that will be followed by data-driven cell type annotation and cell ontology representation by Dr. Scheuermann from the National Library of Medicine. I don't think I said that before. And Dr. Luecken from Helmholtz Munich. And then we'll follow those two presentations with what we're hoping will be a formative discussion with our webinar participants. All of you will be using breakout room format, and we'll explain that to you as we go. We'd like to identify challenges of creating an international consensus lung cell atlas, identify strategies for addressing those challenges, and decide from you as the stakeholders if this is a topic needing further discussion at the ATS Assembly Workshop Project. And then we'll have a brief report back. So as an introduction, the purpose of this webinar is to introduce an ATS Assembly Workshop Project. This has been named a Cross-Country Consensus Lung Cell Nomenclature Project. It's being sponsored by the ATS Assemblies, the Respiratory Cell Molecular Biology, Respiratory Structure and Function, and the Section on Genetics and Genomics. And again, I have to thank the Section on Genetics and Genomics for sponsoring and organizing today's webinar. So this project is meant to be a collaborative of the lung research community. It includes multiple consortia, the LungMap, Human Lung Cell Atlas, the Human Biomolecular Atlas, but it's not limited to consortium members. We really encourage individual investigators to be part of the overall project. Our work product, for the Assembly Workshop Project is a document where we're hoping to provide access to contemporary lung cell annotation and data references, to provide best practices for publication of single cell data sets, to provide a taxonomy or a framework for aligning a common lung cell language, because with a common language, we can move ahead faster in our research and in discovery of treatment for lung disease. And to describe a methodology by which the community can add cells to this lung cell reference. That you are here today says that you're interested in the research, you're interested in the topic, but I'm hoping also that you're interested in this cross-country consensus nomenclature project. And so you can become involved. Your participation here today as stakeholders in the breakout sessions, as I mentioned before. There will also be an ATS workshop on May 20th, 12:30 to 4:30. Unfortunately, the attendance for the workshop is limited. So you know if you're signed up for the workshop already. But then in addition, the overall project depends on task force and working groups where experts in the field get together to discuss lung cell subtypes organized around superclasses of cells, and including our computational experts. So to become involved, just get in touch with any of the moderators or panelists that are at this session. And then we envision going forward that there will be additional workshops, jamborees, and discussions. So keep an eye out if you're interested in being involved. We've summarized what we think are major challenges to this project, at least for today. And these will be the topics of the breakout sessions. And we will put this slide back up. And when we get to the discussion section, we'll ask you to pick a breakout room to be part of. So that's all for me. And without further ado, I'd like to introduce our first speaker, Dr. Malte Luecken. And we'll get his slides ready. I actually, while he's getting that up, I should also have said that this meeting is recorded today, and it will be shared broadly to the community. And we also encourage you to use the chat as we're going along through the talks and through the breakouts so that we can include your questions and your participation. Looks good.
Can you see my screen? Perfect.
Yes, we can.
Thanks a lot, Gloria, for the brief introduction. My name is Malte Luecken. I'm a principal investigator, computational biologist at Helmholtz Munich. And I've been working on data integration of single-cell data sets for the past approximately 4 to 5 years now. And I'd like to kind of share with you a little bit. I'm thankful to be able to start the discussion here with a few ideas on different reporting standards, cell identification, and multimodality towards building a consensus lung cell nomenclature. As a computational biologist, I really like to start off with NASA's motto here: "In God we trust, all others bring data." So for my mind, it's really important to define, when we define consensus lung cell nomenclature, is to do this based on combining many different data sets. For that, we need to reuse data sets that were generated across the community. We need to understand. So that's kind of the first important components that we need to have. We need to understand what's in those data sets to be able to reuse them properly. We need to know how to define cell identity within these data sets as we put them together. And for that, it'd be useful to have best practices. And of course, typically cellular identity is defined based on RNA, but identity is defined by so many more factors than transcriptomics alone. And then there's kind of, do we have standards for incorporating multimodal measurements into the definition of cellular identity, specifically to interpret cellular identity, but also to maybe better define it, what we cannot do in one modality alone. Let me just start with the common standards. Fortunately, as a quite accessible organ compared to others in the human body, there are many data sets that have been published on healthy human lung, but also in various disease contexts. And this is just a kind of an overview of a couple of them that are now a little bit older, maybe, but there's been plenty published also since then, that all give a different perspective of what a healthy human lung looks like, because they have a different experimental designs. And across all of these, we will have different annotations of cellular identity. Some agree and some do not agree. To kind of bring these together to define a consensus, we have to repurpose and reuse these data sets to bring them together to define what cell identity should be. The challenge for that, a central challenge for that, is that collections of data sets suffer from batch effects. If I take data set 1 from a particular study and I look at in a latent embedding the cell types that I have in this data, I will find nice clusters and I'll be able to interpret this. If I take a second study and combine this into the same space, I will typically get separation of cells, and not only by their identity, but also by their batch. And this is due to various technical reasons and the differences in experimental design. I may have used different sample handling techniques, different protocols, etc. But also could be biological reasons. These samples could come from different anatomical locations. Maybe something I would like to identify or keep in the definition of the cellular identity. It would be a lot easier if these kind of overlapped with one another. So we could understand that there is actually no difference between cells from different studies, but unfortunately that's not the case. And especially if we do not know the identity of these cells a priori, it's crucial to have information on the data sets to be able to evaluate whether we have batch effects or these are meaningful biological signals. And for that, we require standardized metadata to understand where these technical artifacts that potentially affect our definition of cellular identity come from. Now, in the single cell workflow, I'm pretty sure I don't need to describe this to you. There are many different steps that each of which different decisions in each of which can kind of confer artifacts or technical differences between data sets. It's quite clearly tissue procurement, like experimental data processes have a lot of effect, as I'm sure many of you know, and optimizing protocols is very important to get the best possible representation of cellular profiles. But also, which is maybe a little bit less focus has been placed on, the computational analysis side can really affect how we call cellular identity based on decisions made. And just to give you a brief example of this, this is a data set which we worked on quite a long time ago and on the mouse brain, actually, using a technology that no one uses anymore for good reason, this iCell8 data. But at the time we had 12 mice, 2 conditions, 5,500 cells. And with hindsight, we kind of annotated these cell types in the best possible computational pipeline we could find, and then looked at all other computational pipelines we'd attempted before to make sense of this data. And here is an example of the simplest versions of normalization and batch correction you could use, because we had batch effects in this data. And you can see quite a mix of cells of different colors on this UMAP. Now, if I look at with increasing complexity, I have quite different results. And you can clearly see that on the right here, this is the processing pipeline we ended up going with. But had I used any of the previous approaches, I would have had, you would have attained different clusterings of cells in these data sets, which leads to different cell identities per cluster, different marker genes. And therefore, also, I would have called these cells potentially differently, just because in some cases I don't have the resolution maybe, or in some cases I misidentify a particular cell type because the processing pipeline differs. And thus the choice of approach that's being used in one data set to call cell types is crucial to understanding and comparing how these cell types across data sets differ. So for that perspective, understanding the choices that were made at each of these steps across the experimental but also computational pipeline is important context if we want to evaluate whether a goblet cell in one data set is the same thing as this defined goblet cell label in another data set. For that, reporting guidelines are important. So to collect that metadata, we need standardized guidelines for how we can report that information. And this is a study which came out in 2020, which Richard, the second presenter today, was part of, and he pointed me towards very helpfully, was a kind of definition of metadata standards, an attempt to define metadata standards for single cell data sets that were presented. This came out in 2020, so technology has evolved a little bit, but actually a lot of the basics, or the kind of the categories that are laid out in here, and this is also where I got this visualization from, are very, very useful. Recently, we've come together within the Human Cell Atlas integration team to kind of define, build on this MinSCE framework as well, to define potentially a few extra categories that confer batch effects in our hands so far to be able to collect data in a standardized fashion. And this all these metadata guidelines go until approximately raw sequencing data sets that are obtained. Then the cell annotation platform has developed its own set of metadata standards to understand where or how cellular annotations, how they were generated and how to compare them. I'll go a little bit more into that metadata in a second. But importantly, there is an aspect specifically of the complexity of kind of computational analysis pipelines that we cannot record in metadata standards. And for that, it's really crucial to be able to share the code that was used to generate these cellular annotations to analyze this data, and the environments for that. So open sharing of code and of the data in the end is crucial to be able to reuse this data set. I want to give you a brief overview over the kind of how these two standards for the MinSE, but also the tier 1 metadata that we generated in the Human Cell Atlas integration team, how they try to describe different decisions that were made and that could confer batch effects. I'll start with the newer version, the 2024 version, which we've recently kind of come to agreement with on. This includes quite a lot of metadata on tissue procurement, because this is a source of substantial technical variation, or also biological variation, which might be misinterpreted as technical variation. It includes information, where was this sample collected, where was it processed, the manner of death, if it's a post-mortem sample indeed, the type of tissue, how the specimen was taken, the preservation method, etc. Then we move towards kind of tissue dissociation, cell enrichment. And here we kind of protocols can differ. So the URL, just to make sure we can kind of look up that protocol, would be really helpful to understand if there are technical artifacts here, but also information on whether cell enrichment was done, whether the sample handling was done in batches or all together, and information on the viability or how well that worked. In library construction, we now have quite, the number of different protocols that are used is shrinking somewhat, although the specialized protocols are increasing, as a lot of the community is converging around various 10x protocols, as well as a few others. And here we just want to understand the assay that was used, the batches that the data was processed in. The same for sequencing, the batches the sequencing was done in, which can also confer a batch effect, as well as which part of the cDNA library was sequenced. And then for computational analysis, the major sources of batch effect are different versions of the alignment software that were used and different decisions in alignment, such as whether intronic reads should be included in the cell counts. To briefly show kind of which aspects are also covered in MinSCE, which were not taken over into the tier 1 metadata we recently had, we recently kind of developed, is information on spike-ins and amplification. And this is because I think protocols have standardized quite a bit since this publication in 2020, as well as information on different sequencing reads, which we have found to not be as critical for batch effect for conferring batch effects, but may be important to specifically reproduce this exact data. So to give you an overview a bit briefly about the challenges, the overview of reporting guidelines. Standard reporting of single-cell experiments is crucial for data reuse to be able to define cellular nomenclature across many different data sets. And each step along this process of generating the data and generating the annotations can confer a batch effect. However, standardization of several things, such as 510x, has simplified the reporting, and we try to capture this in the tier 1 metadata from the HCA. On the other hand, computational pipelines are very complex, and therefore it's critical to share both code via, for example, GitHub or Zenodo, and also, importantly, which is more rarely done, unfortunately, is sharing of computational environments. So only sharing the code does not mean they can be reproduced without knowing exactly what version of what package was used to generate these annotations. So much for the first topic I would like to cover. Once we have standardized data sets, so standardized metadata, standardized information on these data sets, which we know, so we know how the information or how the annotations in these data sets were generated. We need to understand how to kind of renew this identification of cells and to make sure that this is done in the best possible way across these data sets when we repurpose them. Now, of course, cellular identity can be, or characteristics can be defined across different modalities. However, typically, most of these modalities are either not useful to understand cellular identity on the genome level, rarely profiled, or always profiled together with RNA, and therefore also RNA is typically used to define cellular identity when using, for example, single-cell attack sequencing data. Thus, what I'm going to focus on for this part is mostly on the transcriptome. And here, it's, as I kind of showed you before, processing transcriptomic data, the way that is processed is crucial to understanding cellular identity. So a really good important starting point for defining cellular identity is that pre-processing of this is done in accordance with best practices. And there are several resources for best practices processing of single-cell datasets, a couple of which I have contributed to or led in kind of a more Python environment. And then there's best practices also for data processing in R. But either of these, a lot of the steps coarsely agree at least and give fairly good output. And so this would be a really important step to get to the point at which cellular identity can be defined. Now, let's take the kind of the newest version of this, the best practices book that was, I think, published last year. And actually, this is also somewhat of a consensus actually across the data analysis community, or the single-cell data analysis community, that cellular identity or cell groups of cells are typically annotated together. So we use clusters from a graph-based clustering of single-cell data. And this is done via modularity optimization approaches. These are the details on using Louvain or newer now Leiden methods. Importantly, though, we typically do not annotate individual cells because this is comparatively noisy with depending on sequencing depth, larger or less large zeros and dropout rates of genes in cells. And thus, it's more robust to annotate groups of cells together. The challenge, of course, is selecting the right resolution for finding which grouping of cells is a meaningful resolution. When should you go deeper? When should you go less deep? And there are a couple of, there's multiple papers on this topic, a couple of recent solutions that have been proposed in the last 4 years in some cases actually, is kind of more using statistical models to assess whether marker gene distributions are different between subclusters, trying to predict the cellular cluster for cells in one cluster. And if you predict just as likely another cell cluster than the cluster the cells are currently in, then maybe you should be merging those two using a kind of SC-CAF. And then, the best practices book recommendation here is just to, try multiple different resolutions and see what matches, what works best, which is not particularly standardized. But unfortunately, the guiding principle still is at the moment that gene profiles should somehow significantly differ between subclusters. But there's no ground truth final solution for how that exactly needs to happen. Now, if we have clusters, we understand the resolution that seems to work best for our data. The important thing is now to interpret these clusters of cells. And there are different approaches to identify cells going from a cell transcriptomic cluster to a cell type. The simplest case, this is manual annotation, looking for marker genes via, for example, a t-test or more complex approaches, and then comparing these with literature-derived markers in previous publications. We can also learn those we can also automate that process, put those markers into a model, and there's approaches, for example, Garnet, which was published 4 years ago, approximately 4 or 5 years ago, that uses kind of decision models to suggest when a marker is sufficiently high in a particular cell type, and then it annotates in this way. Instead of using literature curated markers, you can also learn which profiles or which genes are important from the data itself. Here you need many more data sets typically. And you then learn based on, for example, logistic regression models as done in Cell Typist here, what are the important profiles that distinguish cells of different type. And you can then use that as a predictive model on new data sets. Or we can use references such as the Human Lung Cell Atlas or CellRef, and use that with the data integration models, use transfer learning approaches such as SCRchess and ScanVI to map new data sets onto a reference and transfer the labels that way. These differ based on kind of literature-based markers as well as database markers, and the level of complexity increases where at the very end we have very explicit correction of batch effects between data sets. But with all of these approaches that are available, what is the best practice approach to define cellular identity? Fortunately, there is quite a nice paper published a couple of years ago, in 2021, from the lab of Gary Bader, where they suggest combining automated and manual annotation to ensure we can leverage the speed of automated approaches, and which work fairly well, especially on a course level, as well as the fine-grained inspection of the data that we need when kind of deciding on especially novel cell types. So as a starting point, they suggest mapping your query data set to a reference to annotate these cells, and some will be annotatable and some will not be annotatable if those cells don't happen to be in the reference, which is very possible. Then use marker genes curated from the literature to evaluate how well that worked, and inspect then differences within these potentially coarse clusters to see whether we can go deeper to annotate subclusters of cells, really taking into account the function of genes whose expression differs between those subclusters to get to a kind of a final fully annotated version. As a final step then, especially when defining newer subclusters or newer cell types, they recommend verifying these through either follow-up experiments, independent data integration, so comparing with other data sets, supported by statistics. Are these clusters only there in one sample or only there in a single technology, for example, which might suggest that these are outliers, or consult with experts in that particular lineage. And so this kind of enables you to go at much larger scale, and much faster speed to combine this kind of fast-paced automated annotation and the attention to detail. However, this assumes we have a reference. What we'd like to do here is to define or understand how to define the reference in a cross-consortium, cross-country, intercontinental, as I think Gloria mentioned, way. So we actually don't have this automated approach available. We're kind of left with manual annotation in that case. But really what it boils down to is this verification, right? How do we, to get as many opinions, to get as high quality consensus as possible, we need to make sure that every step of the way we verify. So we need data integration with multiple data sets, statistical support to make sure that the reported cells or the supported cell identities are not outliers. And we need to consult with as many experts as possible, where it would be great to get as many people's input as possible, as Gloria introduced in the beginning as well. So how do we build a reference? And I'm going to showcase how we built the Human Lung Cell Atlas. Of course, CellRef from LungMap also exists as another reference that is out there at the moment. So here we took 14 data sets, which were landmark studies in the fields, combined these using an optimized data integration pipeline to build a representation of the cellular landscape of the human lung. We could, just me and Lisa, who spearheaded this project, we were just about able to, annotate the different compartments and saw that it wasn't so bad. But the crucial input was given by 6 experts named here, of which I know that at least half of them are on the call today. So who provided kind of crucial input to make sure that these cell identities that were defined based on the clustering of the integrated object was done in a meaningful way. And the important part is that this is a kind of consensus finding approach, that we get multiple opinions, and only through the opinions and the confrontation discussion among these individuals can we define a higher quality of annotation. Now, of course, this is 61 cell identities. This is not the largest reporter number of cells across any study. But therefore, these cells are robust. In a second version, we are currently looking to kind of build a cross-consortium reference combining the datasets in CellRef as well as the HLCV1 that extend to spatial multimodal data. This is a project which Raphael and Martina are spearheading with input from a lot of people also on this call. But how do we kind of get more input from 6 experts? When we did it with the 6 experts before, we could kind of use a Google Sheet and ask everyone to fill out something. And it was very kind of hacky, but it worked. Fortunately, there is now tooling for this kind of thing. So the cell annotation platform is a platform that is specifically designed so that you can kind of upload a data set and everyone has their private little workspaces to give their own opinion. And in the end, this is the idea that, to generate a consensus, every annotation is in the end an opinion, and to combine those opinions on a platform so that you can facilitate the consensus finding. So this works by, on the platform, creating kind of these annotation opinions individually. These can be shared then across a wider community. You can use automated annotation prediction if you like. There are tools for that to get some help to get course overview. But then there are approaches or workflows to help people who are contributing to converge upon a consensus. And in this approach, you are kind of assessing each other's each other's annotation opinions to give that info. For example, if we start with providing a grouping of cells, then an individual kind of labels cells based on the interpretation of data. And then there's a third step to generate one set of annotations, define this kind of metadata, the annotation metadata, the characteristics of the particular label that was given. And there's 10 different fields that are defined here. This is kind of the name, the full name, the rationale, why this annotation was given, marker genes, as well as literature or canonical marker genes, potentially other markers that were used. And then, whether this exists in the cell ontology, and Richard later is going to tell you a little bit more about that. And then kind of in giving information on how was this annotation achieved? Was this manual? Was this automatic? Was this both? What versions of what tool was used to do it, etc. And then when we have this information from enough people, we can start voting. So there can be several rounds of votes, address kind of major disagreements between people. And the way you can disagree is that when you share your annotations, you can either say, oh, I disagree with this person's clustering or the label they're giving. I would like to merge these two clusters or separate them. Or you can disagree with the individual metadata. So you can say, oh, I think the marker gene that was given here is not good. And thereby you can refine that metadata and kind of converge slowly to a consensus. And this is kind of a tool specifically to work on at the moment to work on the transcriptomics of annotating one consensus kind of, or one atlas, if you will, and combining information that way. So to kind of give you an overview over the best practice of cell identification, the best practice for individual data sets is combining automated annotation with manual annotation. But really, to kind of build the reference to do this automated annotation, we need to integrate multiple data sets and to collect expert annotation opinions. And a tool that can help doing this is the cell annotation platform. The challenges that still exist is that choosing a clustering resolution is not trivial, and people will do this differently. And there may be residual batch effects, even in the best integration. So it's probably useful to only annotate clusters which have, which are robust to a certain extent. So they have samples from cells from at least more than one sample, more than one donor, and more than one batch, etc. And then there's still very much a lack in the tooling at the moment, that we don't really have a good way to annotate cellular gradients. At the moment, we just cluster and then put this in a trajectory. But maybe these phenotypes, continuous phenotypes, shouldn't be clustered. And that's something that still needs better methods that are developed. Now that we have standardized ways of reporting metadata and for identifying cells, we have some best practice suggestions. How do we include the additional complexity in cellular identity? And already in 2019, single-cell multiomics became kind of a method of the year in Nature Methods. And this is just an overview over the multi-omic profiling approaches that existed at that time. Now there are far, far more. While current approaches to defining cellular identity is very much on the transcriptome side, this does not have to be the case. We know we can do better. There are tools to do better. And to just give a brief overview, I don't think there are really standards for doing this yet, but there are different approaches for defining cellular identity based on multimodal information. We can, for example, use data integration of paired measurements. This is site-seq data where we measure protein and RNA concentrations at the same time. We can jointly embed these if they're measured on the same cells using approaches like total VI, multi-VI, MOFA plus, and there are many, many others. We can integrate unpaired cellular profiles across modalities. This is what's called mosaic integration, or kind of diagonal integration potentially. The use case is very similar to the first case, only this is potentially more coarse, but much more powerful. We don't need to have done joint profiling. We can combine data sets across different modalities, and approaches that do this is Max Fuse, which was recently published, SC Glue, a little bit older, which has, based on benchmarks, it seems to work fairly well, but they haven't been compared to the others, and SC MoMap. We can also map data sets. So we can use RNA information to represent cellular identity, and then contextualize those cellular identities by mapping information, for example, from spatial transcriptomics pangram on top of these cells, so that we understand how to interpret a particular cluster of cells. You can also do this with Seurat's bridge integration, where you used a multimodal data set to map other modalities like attack on top. And in this case, we can enrich the profiles we get from individual transcriptomics data with spatial information or attack information to really understand regulatory profiles. And finally, if we don't have multimodal information on the same resolution, we can use deconvolution approaches that, for example, if we only have coarse transcriptomic profiles in space, can provide still spatial context for where particular cell identities are likely to be placed and help interpretation. Now, there are no standards here. We have attempted to try to see what the best practice is here. In 2021, we ran a competition at NeurIPS to ask teams to submit approaches to improve how we map data sets, for example, together or integrate multimodal data sets. And we had 172 teams that submitted over 400 submissions. And here, which was really nice, the GLUE team, they won. They could map different modalities together when they're not profiled together. And SC-GLUE came out shortly afterwards, and there's still other approaches in the pipeline. But it's unclear whether that is really a representative sample, because the other approaches, for example, MaxFuse, SC-MOMAT, have not been tested then, and this is a very active field of method development. So I think standards are yet to be stated. So finally, I really want to get to the kind of a brief overview at the end to introduce another tool. If we have this information, where there's many different ways to record multimodal information on cellular identity, where do we store that? How do we log that to make sure this information is kind of standardized in the way that this is kind of stored and made available to the community? And here I want to highlight the REDCap database from LungMap, which is a very comprehensive kind of tool that not only looks at transcriptomics, but really kind of focuses on standardized descriptions of cell identity that range from transcriptomics and markers, of course, also to morphological feature descriptions. So you can use images of cells to kind of enrich that identity, the information that is available on the identity, these protein markers, spatial localization, information on anatomical specifications, and really the number of modalities that can be added is incredibly diverse here. Looking at flow cytometry, but then also contextualizing this with disease, with so I think this database tries to enable a very complex, complete picture of all the context that could be made available to understand cellular identity. So to summarize everything, I think HCA Tier 1 metadata is an attempt to standardize reporting of single cell data, building on existing kind of MIN-SCE approach. We have best practices for cell identification for individual data sets. To do this across data sets, I think is very useful to kind of integrate data using the standardized reporting, and something we're trying to do with HLCIV2 here. To annotate that in the kind of a tool that enables everyone to give their opinions and reach a consensus as quickly as possible, CAP is a very useful tooling. Multimodal data, of course, is missing then at this point. We need to map that on top, and that information can be stored on REDCap in the end. And with that, I need to thank a lot of people. I always like highlighting, making Munich look very pretty here. My lab team recently, and a lot of people who contributed to HLCA V1, to discussions, to a lot of the other things that I presented. And I'm really happy to be able to start the discussion about this. Thank you.
Excellent, Malte, thank you. We're in interest of time, we're going to move right on to Richard's talk, Dr. Scheuermann's talk. Again, encourage you to put things into the chat if you have comments or questions, and again to bring them to the breakouts at the end. Oops. We're doing the talk backwards. There we go.
Yeah, sorry.
Very good. Thanks. No, thank you.
All right. Well, I think Malte did a great job of introducing and really emphasizing the importance of quality control and standardization of the data that we're trying to utilize for building these kinds of organ and tissue atlases. What I wanted to do is try to dive into some of the data and use that as a way of illustrating, I think, some important concepts that Malte touched on in his talk, but just to give you a sense for how the data looks and how it will influence these kinds of decisions. So I'm Richard Scheuermann. I'm the scientific director at the National Library of Medicine here at the U.S. National Institutes of Health. The work that I'm going to present was done in collaboration with several really excellent computational biologists, including Renee Zhang's group at JCVI, Angela Liu and Beverly Pang, and Ajit in my group here at the National Library of Medicine. So I'd like to cover 4 topics during my presentation. The first one is having a discussion about marker genes and different approaches that groups have been using to identify marker genes, and talk specifically about a method that we've been working on to try to develop minimum sets of marker genes that can serve as necessary and sufficient conditions for cell type definition and classification, and what kind of implications the marker genes have for representing this information in reference resources like the cell ontology. I'd also like to talk a little bit about this notion of how do we identify parent cell classes for these cell types we're defining in these experiments, and how hierarchical clustering might be a strategy that's taking a data-driven approach to determine what are appropriate kind of higher level groupings of these cell types we're identifying. And then finally, this notion of let's assume that we develop a reference lung cell atlas that everybody's happy with. How do we incrementally grow the content of that information when new data sets are made available? So first, let's talk a little bit about marker genes. So I just pulled out some examples of marker genes from a number of recent publications and from some computational resources. Malte already talked quite a bit about the HLCA project and this paper that was published from that analysis. And in one of their supplementary tables, they have a list of marker genes, in this case for, say, the AT2 type 2 pneumocyte. There were 3 marker genes that they proposed that represent that cell type. And then for over here for adventitial fibroblasts, there are some different marker genes that were identified, et cetera. And so, HubMap also has their set of marker genes. CellRef came up with their set of marker genes, et cetera. So I think it's worthwhile just thinking about what is it that we mean when we say something is a marker gene for a particular cell type. We have, in the approach that I'm going to describe to come up with minimum combinations of marker genes, we had a very specific objective of what we were trying to accomplish. We think that cell type marker genes are genes that are selectively expressed in cells of a given type, but also that can be reliably used as a canonical characteristic that can then be used to classify them and classify new cells that we might get in a new experiment to determine if it's an example of a cell of the same type. So different strategies can be used to define marker genes. You can do manual curation, which is kind of nice approach to reflect known biology. And this facilitates integration with prior knowledge that might be reported in the literature. And I think HubMap has used manual curation quite a bit to define their marker genes. You can also use selective differential expression analysis. This is a nice data-driven and statistically rigorous approach that's used by HLCA and CellRef. And then there's this approach that we've developed that's designed to identify minimum combinations of marker genes that we would argue could be necessary and sufficient conditions to define cell types that can be used optimally for classification. And on the right-hand side, these are just some uses of marker genes. For example, you can use them as probe sets for spatial transcriptomic analysis. You can use them as kind of a reduced feature space to do data set comparison. And we are also using these marker genes to help come up with semantic representations of cell types and the definitions of cell types that we are incorporated into the cell ontology. So the method that we developed is called NSForest. It's based on random forest machine learning. And essentially the way that this works is that you have your expression matrix and you've clustered it using these kind of clustering methods that Malte mentioned. And then we can actually build classification models for each of the cell type clusters. And as we're doing that, we can actually keep track of the features, the genes, that are most useful in building these classification models. We added in kind of a binary scoring step to re-rank those informative features that enrich for genes that have a binary expression pattern, so they're on in the target cluster and basically not expressed in off-target clusters. And then we can assess how well these marker gene combinations are at classifying cells using a decision tree approach. And then we can use F-beta score or other kinds of summary statistics to quantify how good these marker gene combinations are in classifying cells of a given type. And we've applied this approach in a number of recent publications on a number of different tissues, and it works quite well. And I'll show you examples of what the data looks like. In the most recent version of the algorithm, we decided to modularize the method so that we can actually take any collection of marker genes and assess how well they are in classifying cells of a given type using these decision trees. And so what I'm going to show you now is these other marker sets, how well are they in solving this classification problem? How well do they work? And so I'll show you the results from that. And we basically kind of, through this modularization, now we can actually use any marker gene set to assess their performance. And so we decided to do this using one reference data set as kind of our test case here. And we used the HLCA core reference data to assess how well the different sources of markers could classify all the different cell types that were identified in the HLCA core. And so we collected marker sets from HLCA, from CellRef, from Azimuth, from HubMap, and from running the NS-Forest algorithm on the HLCA data. And the trick here is that we have to be able to match cell types across these different data sets. So we need to be able to match the cell types that were identified in the CellRef experiment to the HLCA data that we're going to be using for doing this comparison. And so we did that by kind of a manual curation, trying to match names, trying to match cell ontology annotations, looking at the overlap between marker genes to try to get, a best case matching of cell types across these different data sets. And so once we were able to do that, then we could assess classification performance of the different marker genes on this common HLCA core data set. So these are dot plots that show the expression pattern of the markers from these different sources. And so just to orient you to what these plots are showing is on the x-axis here, these are the different cell types that were defined in the HLCA data set. And then on the y-axis, the vertical axis here, these are the different marker genes from these different marker gene sources. And so ideally what you would like to see is a really nice clean diagonal pattern of expression of marker genes that are really restricted to these individual columns of cell types with very little off-target pattern, in these dot plots. And so this is what you see for the NS-Forest markers. These are the HLCA markers that were reported in their paper. It's also pretty good, a little bit more off diagonal expression of some of the markers, but all in all, they're not too bad. The same with the CellRef. The azimuth markers there's a lot more kind of off diagonal expression. And I think this is because they have a much larger number of markers for each cell type that they're using. And then here's the results from the ASCT plus B tables for HubMap. And I should just mention that I think that, all of these marker sets are useful, and they've been developed probably for different objectives, for different purposes. And so depending on what you're using the markers for, you may want to choose one or the other of these marker sets. But I think for us, for this notion of building kind of these minimal marker sets for classification, we think that the NS-Forest approach is giving us kind of the characteristics that we're looking for. So this is now looking at these kind of statistics that measure the performance of these marker sets using these decision trees as a way of assessing their performance. And so typically you can measure two things, precision and recall. Precision, and the equations for these things are down here. So precision is basically looking at the proportion of true positives divided by all the positive calls. And you can see that all of these marker sets actually do really a pretty good job in the precision metric. But where we see a big difference is in the characteristic of recall. And in recall, the key difference is that the denominator includes the number of false negative calls instead of false positive calls. And then this F beta statistic is kind of a combination of precision and recall. And so it kind of is looking at, we want the best of both worlds. We want to have high precision, we want to have high recall. And so we can use F beta as a way of combining the two to get a summary statistic that gives us, that's looking at kind of the best of both worlds. And so what we see here is that because of the way that the NS-Forest algorithm is picking out marker genes, it tends to do a good job in both the precision and recall evaluation. So this is now a way of looking at comparing these results from the different marker collections. And what we're doing here is in the first row, we're looking at the F beta statistic. And in the x-axis, this is the NS-Forest markers. And in the y-axis, we have the markers from these other sources. And what you can see is that for F beta, NS-Forest tends to give higher F beta statistics in comparison to all four of these other marker set collections. For the precision metric, we see that, sometimes NS-Forest gives better precision, sometimes the other marker sets give better precision, and we see that that's the case for most of these other marker sets. But then there's also this group down here, which for whatever reason, NS-Forest is better at defining based on precision. And then down here is recall, and this kind of reflects what we're seeing for the F beta statistic in general. And this is just some examples that kind of provide an explanation for what's going on here. So I've just picked out these three different cell types from the HLCA data, and here are the different marker sets, and these are all these different kind of statistics that we can calculate. And so I think the biggest difference here is that many of these marker sets are really good at kind of distinguishing between true positives and false positives. So let's, for example, here for the AT2 cells with the ASCT plus B markers, you see we get about 8,000 true positive calls and only 56 false positives. And that's why you get this really high precision measurement. But the problem is that this marker set combination is very stringent, and so you end up with a lot of false negative calls. And that's why the recall statistic is relatively low. And so with NS-Forest here, you see that we still get a lot of true positive calls here, but the real benefit is that we're not getting very many false negatives. And that's why we get both good precision and recall using this technique. And then finally, the other thing that I'll just mention is that the number of marker genes from these different sources is different. And NS-Forest is specifically designed to come up with this minimum set. And so just to kind of summarize, we think that this algorithm based on machine learning really maximizes the retrieval of true positives while minimizing both false positives and false negatives. And so we think that it has some real value for a lot of the downstream applications. So what are the implications for cell ontology representation? So the cell ontology is an ontology of normal cell types. There are about 3,000 classes as of this month. It's it's built as a parent-child hierarchy of cell types where the parents and children are connected by ISA relationships. The nice thing about the cell ontology is we can also capture developmental relationships in developmental trajectories using develops from relationships. But I think one of the challenges that we're running into in being able to use the cell ontology to help us represent the data that's coming out of these single cell transcriptomic experiments is that in the current ontology, many of the definitions don't include markers. And when they do include markers, they're usually protein markers. And so, in these experiments, we're defining cell types based on transcriptional markers, and it can be challenging to determine whether this cell type cluster we found in this single cell transcriptomic experiment really corresponds to a cell type that's already in the ontology. And so I'll show you kind of some examples of that. So several of the lung data sets have mapped their cell types to cell ontology terms. So again, this is an example from the HLCA data set. So there are some matches to existing CL terms that seem to be correct. So for example, here in the HLCA annotation, this cell type cluster is labeled as a classical monocyte. And here in the cell ontology, there is a term classical monocyte. And so just based on the terminology, we would say, okay, well, that's a match. You have things like mast cells and NK cells and plasma cells and B cells. We're fairly confident that those things are really good matches between the cell type cluster in the data and the cell representation in the cell ontology. But then if you come up here, you see that in this case, we have that all of these different kind of alveolar macrophage subclasses are all mapped to the same cell ontology term, alveolar macrophages. And so obviously this can't be an exact match between these cell types and this class, because, if this were all kind of appropriate, each individual cell type should have its own unique representation in the cell ontology. So that's one issue. Another issue is we have a situation like this where this is labeled as DC2 in the HLCA data. It's been mapped to the CD1C positive myeloid dendritic cells. Is that really a correct match? It's hard to tell just based on the label. And another example is mature and proliferating lymphatic EC cells. So those are down here at the bottom. Again, they're mapped to a single term in the cell ontology. And so we're not making a distinction between mature endothelial cells and proliferating endothelial cells. So these are some of the issues that we're running into as we're trying to map these cell type clusters to the cell ontology. And I think what's happening here is that in some cases, we have an exact match between things, and in other cases, we're not. And maybe this is a match to a parent cell type that's present in the cell ontology, and there isn't one that's a map to this more granular cell type. But that's not clear from the annotations that are being provided. And so actually the ontology community has recognized that this is an issue, and they've come up with a strategy for being explicit about when we know things or when we suspect things are an exact match and when they're not an exact match, and what other kinds of matches are we seeing. And so this approach is called the simple standard for sharing ontological mappings. And it allows you to explicitly state whether you think something is an exact match, whether it's maybe a match to a broader cell class, or whether it's a related match. We think these two cell types are related to each other. We're not sure exactly how they relate to each other. And so now we can go back to these matches here, and we can start to now add in this explicit representation of whether we think these things are precise matches or not. So now down here, again, we have our plasma cells and B cells. We can label these as being exact matches. But now we can come up here and we can say, okay, well, we think that these different alveolar macrophage subtypes are a broad match to this parent cell class of alveolar macrophages. And so now we can be more explicit about what we know and what we don't know as we're doing this mapping between the cell type clusters and the cell ontology terms. So we have some high confidence exact matches, especially for lymphocytes. The proliferating cell types are not currently found in the cell ontology, so this is something that we can request consideration for adding. And I think another of the challenges is that most of the CL definitions lack these transcriptional markers, and many are problematic for other reasons. And so in that case, we might use something like related match until we kind of get those issues resolved. But then we can actually propose, okay, so how can we use this to incorporate this information into the cell ontology? So in situations where we have an exact match, so here's an example of goblet nasal is probably an exact match to nasal mucosa goblet cell in the cell ontology. There's an existing definition for that cell type, but it doesn't have markers in it. And so now what we would propose is that we can take this information and we can add new information to the existing definition in the cell ontology to say that these cells selectively express this combination of transcripts in human lung. And so now where there is an exact match, we can start to augment the cell ontology and add in these transcriptional markers to be able to kind of expand the description of these cell type characteristics. And then if we know that there's an equivalent cell type, say in mouse lung, we can also say that, well, the cell type expresses marker 3 and marker 4 transcripts in mouse lung. And so we can continue to augment the content of the cell ontology in this way. When there's a broad match, what we would say is, okay, here's club nasal is a broad match of club cells. We can request a new term to the cell ontology, a nasal club cell, and we can provide a definition, a club cell located in the nasal epithelium. These cells selectively express this combination of transcripts. And so in this way, we can add content to the cell ontology when there's an exact match, and then propose new terms to be added to the ontology when there's this broad match. When there's a related match, we would have to have some discussion about how we would best take care of that. All right, so I want to touch briefly on this notion of how do we determine what is a parent cell class for these cell types that we're defining. So again, this is an example from HLCA. So here for AT2 cells, we have this notion that there are these different levels of parent cell types that we can represent hierarchically. So AT2 cells would be a type of alveolar epithelial cell, which would be a type of epithelial cell. And so we have these kind of different levels of annotation for the different HLCA cell types. So the other thing we can do with this data is we can take our data and we can generate hierarchical clustering results that captures the relatedness between the cell types that we're defining in these single cell data sets. And that's represented in these kinds of dendrogram representations. And so if we take these annotations for these higher level groupings and add that to this kind of dendrogram representation, at this annotation level 1, we see that there's a pretty good match. So here are these immune cell types of macrophages and dendritic cells and lymphocytes, and they match the structure of this dendrogram pretty well. And here we have the stromal cells that include fibroblasts and smooth muscle cells. And over here we have epithelial cells that include the pneumocytes and goblet cells and club cells, et cetera. And so these annotations match the structure of these dendrograms pretty well. But now we're seeing that if you look at the endothelial cells, we're seeing that there's some differences. And we have these two clusters, one that seems to include the lymphatic endothelial cells, and one that includes the blood vessel-derived endothelial cells. So now when we go down to the next level down, things start to break up a little bit more. And so you see here that things that are labeled as myeloid cells are actually in 3 different branches here that are kind of interspersed with other cell types. And we see the same thing of interspersion between fibroblasts and smooth muscle cells, and over here between airway epithelial cells, alveolar epithelial cells, and submucosal gland subtypes. So to me, I think what this raises is a question about, are these different higher level annotations, are they really reflecting the relatedness between the cell types? Are they reflecting the developmental origin of these cell types? And is this something that maybe we should look into a little bit more carefully, just to see, if maybe we're learning some new knowledge, new biology about how these different cell types are being generated? So another strategy is to actually use the structure of these dendrograms as a way of defining the parent cell classes. And so you can kind of cut the dendrogram at specific places and derive these groupings based on the structure of the dendrogram. And if you do that, then you might propose that this is actually an alternative classification of these different cell types into parent cell classes. So I know that I'm probably out of time. So let me just very briefly talk about incremental growth of lung cell types with new data. So the idea is that, let's say you have a reference atlas, you have a new experiment, you want to basically integrate the data from that new experiment into that reference atlas. How do you go about doing that? There are different approaches that have been developed to do this different type of matching. This is an algorithm we developed called FR-Match. I'm not going to go into the details because of time, but there are different ways that groups have developed to try to match data sets to do this kind of thing. So this is an example where we applied our algorithm to match data sets from 2 regions of the human brain, the primary motor cortex and the middle temporal gyrus. And we wanted to know, are the neurons in these 2 different brain regions the same, or are they different? And what we found, I think, is really interesting from a biological perspective. So here we have the middle temporal gyrus on the vertical axis and primary motor cortex on the horizontal axis. And what we found is that the GABAergic inhibitory neurons are conserved across these 2 brain regions. They're the same types of cells we're observing in the 2 brain regions, whereas the excitatory glutamatergic neurons are not conserved. They're distinct in the different brain regions. So, and we had a chance to, as of yesterday, this is very preliminary mapping, but we did this to try to map the cell types in the HLCA core with the cell types in the CellRef reference. And I'm pretty actually happy with this first kind of preliminary result, where we're getting matching that seems to make sense between these 2 reference atlases. And one of the things that's kind of interesting is down here, these spaces here where we're not getting a mapping, these correspond to the club cells and the goblet cells from the nasal cavity, or the multiciliated cells from the nasal cavity, which are present in the HLCA core, but are not present in the CellRef. And my understanding is that that makes sense based on the source of the material that was used in CellRef. But anyway, I think this is probably what we're talking about is, and I think Malte talked a little bit about this in his presentation, can we come up with a lung reference? And it looks like maybe the best strategy might be to take the HLCA data and the CellRef data, integrate them together using these different kinds of tools. And then as new data come along, we can compare and incrementally add to the cell reference, the lung reference, when we identify new cell types. And we can incrementally add information about the details of these experiments for cells that are already existent in the cell reference. So with that, I'll stop and just acknowledge my collaborators and the funding sources, and thank you for your attention.
Thank you, Richard. A lot to digest in both of those talks and integrate between those talks. Martin, do you want to carry us on?
Yeah, I'll take over. So thanks for both speakers for very good, intriguing talks. I'm still processing some of the things Richard said, and we'll probably keep doing that for a while. But anyway, we're now breaking up into breakout rooms. We're still with a good 75 people. So I think if we have 5 breakout rooms of around 15 people, that should be fine. You can see the challenges here. These are the topics of the 5 breakout rooms. In principle, we would allow you to pick the breakout room that you would prefer to go to and to join the discussion. But also, we would like to ask you to be a little bit considerate of the total number of people in each room. So if there's a room that's already very full, try to find a room that might be, still has space for you. In principle, we'll have a 15-minute breakout, I think, considering the time, and then reporting back from the breakout rooms. And the goal of this breakout is really to take the challenges here that you can see in your screen, and then discuss strategies and approaches, how we can resolve some of these challenges, which will feed into the document and maybe into the workshop we'll have at ATS, where we will follow up on those discussions. So with that being said, can we somehow see the breakout?
Yes.
I don't see them in my screen.
Okay, so I just opened all the breakout
Should be
rooms, so
coming.
you should see a red button on the bottom right of your Zoom screen that says breakout rooms. If you click that, you'll be able to join one of the 5 breakout rooms.
Okay, one reminder for all the moderators of the breakout room, please press the record button in the breakout room as well, so that we'll have the transcripts of those discussions, because I think it's going to be very important for us moving forward.
I'd also encourage the speakers in particular, if you have answers to the pieces that were put into the chat, you might go ahead and add them into the chat if we don't have time to talk about them. So see you in the breakout room. Yes, I just want to thank everybody who's still in the meeting, who's been in the meeting. I hope that this was useful presentations. They certainly were useful for me and interesting. I thank Malte and Richard for presenting them. We didn't have a lot of time in the breakout sessions, but it was easy to get discussion going, at least in ours. So I know that this will continue for quite some time. We'll have, as we said before, multiple opportunities, hopefully, for people. And just, we wanted to start the cross consortia and investigator discussions. So I think we have some good ideas going forward. If anybody has specific workshop topics, they want to make sure that we discuss at the workshop and in the subsequent meetings, please email any one of us and we'll take it forward. Anybody have a final comment? Thumbs up. I'm seeing thumbs up. Perfect. Excellent. All right.
Thank you,
All right.
Gloria.
Thank you.
For sharing this.
Yes, and thanks again to the section, Genetics and Genomics.
Yeah.
This was terrific. Thanks again.
Thank you.
Thanks, everyone.
Bye.
See you at the ATS. Bye.
Thank you. Bye.